Namaskar. Today we'll study the positive regulators of cell cycle. So before that, what is cell cycle? The stages which a cell undergoes from one division to the next division are called as the cell cycle. And as we know, there are two stages or two phases: interphase and M phase or mitosis phase. And interphase consists of G1 phase, S phase and G2 phase. S phase is DNA synthesis, G1 phase is the pre-DNA synthesis and G2 phase is the post-DNA synthesis or preparation for mitosis. And after the cell division, the cell may remain in the G0 phase or the resting phase, and for some cells the cells are permanently arrested in this G0 phase, and those cells are called as permanent cells. For example, skeletal muscle fibers, cardiac muscle fibers, nerve cells. And for other cells, there are some positive regulators of cell cycle which will help the cell to move from G0 to G1 phase, from G1 to S, and from S to G2, from G2 to M, like that. And these positive regulators of cell cycle are called as cyclins and cyclin-dependent kinases. Similarly, there are negative regulators of cell cycle whose job is to arrest these positive regulators so that they will stop the cell division and the cell will undergo apoptosis. Today we will discuss about the positive regulators of cell cycle. And as you know, the positive regulators of cell cycle are known as cyclin and cyclin-dependent kinases, and the cyclin-dependent kinases are inactive and they become active when they bind to the cyclin and form a complex, cyclin CDK complex, which is active. And this can become inactive by phosphorylation, and again this is a reversible process. The phosphorylated cyclin CDK can again become active by dephosphorylation by the action of the phosphatase enzyme. The CDK inhibitory proteins, they actually bind to both cyclin and CDK in this complex and distort the active sites of CDK, and now the new complex will be the P27 cyclin CDK complex, which is inactive, so they cannot progress the cell cycle. So now let us discuss in detail about the cyclin and CDK. So cyclins control the progression of the cell cycle. They undergo a cycle of synthesis and degradation in each cycle, so they are named as cyclin. And there can be two types of cyclin, G1S cyclin and G2M cyclin. And you can see here G1 to S and G2 to M. Major two checkpoints are there. So G1 to S and G2 to M. These the cyclins mainly they help in progression from G1 to S and from G2 to M. And so we can have two types of cyclin, G1S cyclin, G2M cyclin. G1S cyclin is essential at G1S transition, G2M cyclin is essential at G2M transition. And three cyclins, Cyclin A, Cyclin D, Cyclin E are coming under G1S cyclin. Cyclin B is coming under G2M cyclin. Similarly, we can have cyclin-dependent kinases, a family of protein kinases which are inactive and becomes active after binding to the cyclin. And there are different CDKs like CDK1, CDK2, CDK4, CDK6. And each of these cyclin specifically bind to different CDKs and form complexes. So let us see. Cyclin A can form complex with CDK2 and CDK1. Cyclin D can form complex with CDK4 and CDK6. Cyclin E can form complex with CDK2. Cyclin B can form complex with CDK1. So accordingly, the CDK inhibitor protein P27 inhibits the cyclin A CDK2 or cyclin A CDK1 complex. P16 inhibits cyclin D CDK4 or cyclin D CDK3 complex. P21 inhibits cyclin E CDK2 complex and cyclin B CDK1 complex. So you can see here the cyclin D CDK4 or 6, cyclin E CDK2, they regulate the transition from G1 to S phase. Cyclin A CDK2, they regulate the S phase, DNA synthesis phase. Cyclin B and CDK1 complex regulate the progression from G2 to M phase. And they actually decrease the cohesin and increase the condensin. And what is that? So you can see here, these are the cohesin molecules and these are the condensin molecules. So in mitosis, the prophase, there is chromatin condensation. So they increase the condensin and decrease the cohesin, means they prepare the cell for the mitosis. So that the cell will undergo mitosis and there will be condensation of the chromosomes. Then in this way, these positive regulators of cell cycle act. In the next video, we will see how the negative regulators of cell cycle act. Thank you.